at this point, I'm going to introduce our first speaker, and our first speaker is Andrew Page from Quadram Institute Biosciences. He is the head of informatics there and has been heavily involved in SARS-CoV-2 genome sequencing and analysis for the past 2 years, both at local, national, and international scales. So Andrew's going to set the scene, I think, hopefully, for why SARS-CoV-2 genome sequencing is a useful endeavor and some of the information we can extract from sequencing. So Andrew, over to you. Thank you very much for joining us.
Thank you very much. Yeah, I'm just going to set the scene and show you what you can get out at the end of this course, the kind of things you could do with all the data you're going to generate, and whet your appetite a little bit. So my group, as Nick said, has been involved in a lot of different sequencing. We sequence thousands of samples every week. Last year, we did 65,000 samples. And hopefully we will run out of samples at some point because there'll be no COVID anywhere. But for the meantime, we're sequencing away. So I'm going to just give you a few little case studies of where we found the data to be useful, and at a variety of different scales, from just individual hospitals or care homes all the way up to, national scale. So we had a look in the first wave just at our local little area. So that's about 1 million people in one little part of England. And we sequenced everything we get our hands on, regardless of viral load or CT. And we ended up over 50% of genomes being sequenced, which is quite good. And what we found was that, okay, well, we didn't get 50%, sorry, it was 41%, but we found we actually reasonably well matched what was coming into hospitals and getting a lot of samples sequenced. And we're using Arctic, which you'll hear about a lot more later. And then depending on what type of sequencing or turnaround times, we could batch things up in large batches and do thousands of samples of Illumina, or do nanopore, say, for smaller batches where we need a rapid turnaround time. And with Illumina, we've now scaled our methods up, and we can do about 3,500 samples in a single Illumina run, which brings down the per sample cost quite substantially. Okay, so the first bit of analysis we did was we just looked at, what's out there. And we found that in the early stages of the pandemic, there was a lot of different lineages knocking around. Mutation rate was about 2 or 3 SNPs per month. So that's quite an interesting thing. And for a lot of the pandemic, that's the mutation rate that's been observed elsewhere. And, a lot of stuff circulating locally. These are more finer grained in the early stages of pandemic. So 100 UK lineages, which is basically 100 different clusters we observed within one small area. So that's kind of cool. And those can be used for doing outbreak investigations and for digging into data. So one thing we found was with care homes, care facilities, we were asked to look at one care home. A lot of people have passed away. And when we looked at that, we found it was an uncommon sublineage with a very distinct single SNP. And then we just overlaid all that data, where is this sublineage, where is this cluster found within the local area? And then we just overlaid this on a map, and we saw that there's, hotspots within this little region. And all the hotspots corresponded to individual postcodes, like zip codes. And it seemed to be in very, very old people who were over 80. And also people close enough to one major city. And so then we dug into it a bit further, and we found actually it wasn't just one care facility, it was 6 care facilities, all with this very distinct sublineage. And what we found actually was that it seemed like these care facilities were cross-infecting each other. And that's important because a lot of restrictions are put in place, stopping visitors coming in, testing people before discharge from hospital, this kind of thing. And it seemed that actually he was getting in the back door. It was probably staff or professional visitors moving around, passing around between these care facilities. There was one or two potential cases of hospital-acquired infection, but given the number of cases in care facilities, this was probably not the main driver. As one example, we had a small little town of about 5,000 people with 2 of these care homes. And while there are lots of different lineages circulating within the local town, there's only one lineage within the actual care facilities in those towns. And it's important because actually 40% of the patients died in one of these care facilities. So it's, the mortality rate was quite high, depending on the patient population. And we tracked this till the very bitter end. Luckily, the lineage went extinct. The changes that they made locally were to discourage any movement of staff between care facilities. And we hope that this would reduce cross spread. And everything died out. Unfortunately, it was because a lot of people passed away, but the lineage went extinct, and we're confident in that because we never saw it again, and we never saw it anywhere else in the UK or the world. The next case study we had was we had a large poultry factory. So a person turned up in hospital, severely ill with COVID, and when they took a case history, they found actually they worked in a chicken factory, and in the cutting room, no less. So you've got a lot of people in one environment, actually hundreds of people. And so it made people very worried. So they went and screened the entire factory, and we got some of those genomes, and we sequenced 27 of them on using nanopore. And in under 24 hours, we were able to identify that actually these are all the same, 100% identical, or maybe one SNP in a difference, but basically all the same. So the whole outbreak was related. When we went and looked at GISAID to see where could this potentially have come from, first of all, we didn't find any other cases like this in the UK, so this lineage was not in the UK. And most recent common ancestor was actually seen in Portugal. And an important thing to note is actually a lot of the workers in this factory are Portuguese. Of course, the public health teams went in and interviewed people, and no person reported any travel history out of the country or any family member having a travel history. So we don't know what went on there, but potentially these are low-paid workers, and they didn't want to have to lose money by having to self-isolate, and maybe they just went straight into work. We monitored this very specific lineage because it's quite easy to identify. It's not seen anywhere else. It's got unique mutations, and it actually spilled over into the community. And what happened was it went not just to one factory, but to multiple factories. It went in the local hospital, a non-COVID ward got infected, and some towns had quite a few cases. So yeah, it spread to 2 other poultry factories, causing very large outbreaks, because again, this particular part of the UK has a huge number of poultry production, and yeah, it caused a lot of problems. And what we found actually was that many people were employed by agencies, third-party companies, and the staff were being moved between factories. So one factory would close down because of COVID, and then some of the workers would be moved to another factory, which would lead to new introductions. And like in the third factory we looked at, actually we could tell there were multiple separate introductions just by the mutations that had accumulated at that point. So this, was actually quite useful information for the public health teams. Another interesting thing was that a 4th factory with mass outbreak was totally unlinked, and that actually calmed everyone down. So the interventions here were that they tried to stop workers being moved between factories. And actually this lineage, after about 5 months, it, kind of knocked around, but didn't cause any more large outbreaks and became extinct. So it didn't spill over to the rest of the UK. All we had was one case popped up in one place elsewhere, and that was it, which is quite interesting. Measures actually stopped it in its tracks. Another little case was we were asked to, we've got lots of cases in one particular hospital. Is it all part of the same outbreak? Is staff spreading it? And the answer is actually no. And it allowed them to rule out nosocomial infections, and it's just people were being infected from the community, and it was just kind of free flow of lineages in from the community. And that's important information. It means that your infection control measures are not really the problem. Okay, so fast forward in the UK over the winter or autumn winter of 2020, which is a very long time ago. We had a lot of cases, and so Christmas and New Year's was cancelled. So we were asked to look at another hospital outbreak, and the interesting thing about this was that I'd flagged where that big arrow is a few weeks before the peak, that you've got a problem here. And this was identified because this hospital, these are all cases from hospitals. So, and we sequenced everything from this hospital, every single positive. And so this hospital had a fair few cases coming in, and we identified actually that some of the lineages we were seeing, and some of which were quite distinct, were not seen in community, were in community sequencing. And this could have just been a delay or a lag, but it was more than a coincidence. We thought that these are all just one hospital were seeing this particular lineage. And so the health protection team and the infection control doctors went in to have a closer look. And what they found actually was the red line was caused by staff, and the yellow line, which is a different lineage, was caused by patients. And then the blue line was just, that's the alpha variant coming in the end, just to scupper everything. So we identified that there was a problem before it became a huge problem, and it resulted in actually the staff outbreak, which is a big peak. We identified that earlier, and then it spread to 11 wards. So staff moving between wards were spreading it throughout the hospital and then infecting patients. And this was then used to better inform the infection control measures. So what they did was actually tested staff before moving them between wards for shifts. And they also brought in rapid PCR testing on site within the hospital so that they could actually, get information back that could be usable fast. And that ended that outbreak, but only after it had spread everywhere, unfortunately. Then another lineage was quite a different type of wave, was ever increasing waves. And this was patient spread. And that was, one patient moved from one ward to another. And then, it would spread, but it would spread slowly because patients move slowly, staff move fast. And then they brought in testing for any patients moved anywhere within the hospital unless they were COVID. So that's quite interesting that we're able to deconvolute those, and actually, because we're doing sequencing every week in near real time and getting the data back to the medics, they could take actual real action, which had a real impact. The next thing we looked at was actually prisons in one region. And there were quite a few outbreaks, and some of them were persistent. So one big prison in a city, it had two big outbreaks mirroring the community. And, they pop up on a wing, and then, they'd have mass asymptomatic testing, and they'd try and stop it. And then more would pop in, but it was more like a free flow of lineages from the community inwards and outwards. And we saw some going out as well. And we saw also some people in the local community who were definitely not prisoners. So, women in the community, and these were male prisons. And so, we could tell there's a bit of spillover, but not too much, thank God. And then we looked at a few other prisons, and we found actually they're all unrelated. So there's no spread between prisons despite prison transfers. And some of them had very different demographics, like say a single case, others, had big outbreaks. So interesting stuff. So we went and did, we took a look at different care homes around the world, and actually found that, genomics is quite useful. And it doesn't, well, at the time it was wild type, so it didn't matter what lineage you had, mortality was high. And also we found that bioinformatics and sequencing methods were actually often not necessarily applied the best. And, there's a couple of studies where people made very, very simple mistakes. And, Nick mentioned Delta Cron, so I'm sure that'll be discussed later. So, that seems to be another classic lab mistake. We also looked at importations into England over the summer, and people coming into the UK who tested positive were then genome sequenced. And looking back, it took a huge amount of work. So this is months later, retrospective, but they found actually that you could see the early signs of importations through different routes that maybe were unexpected, or maybe totally expected. Basically young people going on beach holidays, coming back to the UK. There have been this mantra within certain sections of the press that, maybe it's immigrants or whatever, bringing in infection, but actually no, it was all self-inflicted, unfortunately. And this preceded the rapid rise in local cases by a few weeks. You can see the early sparks, and then things took off. We've done a lot of work with we've had a lot of collaborations with people internationally where we've shared expertise or we've helped each other, and that has led to a few different studies that we've helped with. So in Zimbabwe, we've looked at a few different waves there. And in the first wave, they found, things were very closely monitored. And they found, there are different distinct phases of the first wave that they had in Zimbabwe, where people were maybe in government quarantine, first of all, travelers, bringing it in from far away, like Europe and America. Then you had kind of a second phase, which was people in quarantine, hotels, spreading it amongst themselves. And then finally, kind of the third phase was just community spread. So it obviously taken hold, and there was not much you can do, but that did help to inform public health policy. Zimbabwe, also being right beside South Africa, did see a lot of similar things to South Africa at similar times. And so we looked shortly after the Beta variant was identified, and it was unfortunately dominant within Zimbabwe in about a year ago in January. But to try and speed things up, so at this time, samples were sent from Zimbabwe over to the UK. We did send over MinION equipment, sequencers, and we did training over Zoom and WhatsApp. That meant that when the Delta variant came along, they were able to go and do their own sequencing and identify Delta in country much faster than could be done, by sending sequences abroad. And unfortunately, Delta then took over in that country. We also looked at Pakistan and helped them. We took samples from one city and identified, actually, it was all the Alpha variant. This was at a time when Delta was, picking up in India next door. And we could also tell that most of the importations appeared to be related to UK. And there is a very strong connection and a lot of flights between the UK and Pakistan. So unfortunately, the UK probably seeded Pakistan. We looked at Lebanon, and this is the final little case study. So in Lebanon, they had different phases. So in 2020, actually, when other countries, had maybe B1177, the Spanish variant, there was no dominant lineage actually in Lebanon at all in 2020. It was when the Alpha variant came along that within literally a couple of weeks, you can see here in the little figure in the top corner, it just took over. And that's with them imposing travel restrictions. And what happened actually was that we could tell from the genomics data in GISAID that it appeared to be that they weren't infected directly from the UK, but they were infected through third countries. So particularly countries where they had large expat populations. So it came in the back door, basically. So travel restrictions don't necessarily work unless you ban absolutely everyone and whatnot. So just that has been borne in mind. The interesting thing is that there was not consistent sequencing or surveillance over the whole time. So the first signal that Delta was in Lebanon actually came from the Gambia, from their sequencing program. And they identified travelers from Lebanon in their sequencing data who tested positive. They sequenced them, and then they could tell actually, Lebanon clearly has Delta. So surveillance, efforts in the Gambia did a good deed there. We've been helping out finally with setting international standards. So sharing metadata is quite important because a lot of the international studies are only made possible by sharing data. And we have to share it and use the same kind of terms. And finally, if you want to learn more, we do a little podcast and we have about 20 hours of talking about SARS-CoV-2. So check it out on the Microbenefit podcast. And of course, plenty of people to acknowledge. Thank you very much.
Thank you very much, Andrew. I think that really is a great motivation for some of the information that can be gleaned from looking at the evolutionary relationships between sequences in an epidemic scenario. It would be great to have some questions in the chat. So I'll give you a few seconds to think of one for you. I'll ask a question first, I guess, while we're waiting. I definitely want to see one question before we move on to the next speaker. So I guess my question is with Omicron, we've seen absolutely extraordinarily high kind of degree of transmissibility and dissemination globally of that variant, and that variant actually dominating certainly in the UK and in many countries in a very short amount of time, a month or so. In this kind of scenario of a very, very apparently very high transmissible variant, is there still a role for this kind of local scale genomic epidemiology that you were presenting? Or, and if there is, where could or should that effort be focused? Because clearly, it's almost impossible to sequence everything because there are just so many cases, and you require a huge testing infrastructure and a huge amount of money. So where is that, if that effort's still worth doing for something like Omicron or future variants, where is that effort best targeted?
I think actually the best place to target it, well, two places, is people who are severely ill, so turning up in intensive care. You should probably sequence those in case there's a particular variant that is causing severe illness. And also then just community surveillance. So there's a project we're involved in the UK called REACT, and they post out test kits randomly to people in 100,000 test kits every month. And then we sequence all the positives. So that gives you a true reflection of what is in the community, how prevalent is it? And are people symptomatic? Are people, vaccinated or whatnot? And, it gives you an unbiased survey of what is out there. And I think that's what we should probably go towards in the future, rather than just trying to test everyone and sequence everyone. It needs to be much more targeted to give a much more informed decision-making.
Yeah, that makes sense. So a combination of kind of broad scale random surveillance of the population, and then a focus on severely unwell in hospitals is where if you had limited resources, which obviously we all do, that's where you'd focus. Absolutely.
And I'm sure the world will probably go towards that quite rapidly, as things gear down and money gets rationed.
Is there an argument for focusing as well on particularly vulnerable populations where perhaps they're not protected by vaccines? And you mentioned the care homes as a potential area. Is that another place to look at as well?
Yeah, vulnerable populations, of course, but actually with care homes, we're finding that the mortality rates are very, very low now. We don't know if it's because just all the more vulnerable people have passed away, or if it's just because the vaccines are just incredibly effective and there's a very high vaccination rate and very high third jab rate as well. But it's actually really, really low numbers at the moment, thank God.
That is good news. Okay, lovely. We've got some questions in the chat. Okay. So let's start from the top. Question from Amir. In the first case study, the number of genomes was more than the number of patients. is that due to reinfections or infection with different lineages at the same time? Or
yeah, so some people were repeatedly tested, and in some cases we had like people were tested like 6 or 7 times, and we could still get genomes out of them 2 months later. And then some people maybe were immunocompromised, and they had a high viral load within their samples, and they just weren't clearing it. No symptoms, no nothing, whatever. It was just percolating away. And these were particularly heavily checked. I think there was some changes then thereafter, so that you wouldn't go and retest people for like, I think 90 days. But at this time, these were severely ill people, and they were just testing them, every few days, unfortunately.
yeah, I mean that kind of sequential sequencing of longitudinal sequencing of patients can be quite an interesting, can't it? Particularly in the situation where patient doesn't clear their infection quickly. So, in particularly immunodeficient patients can reveal interesting information. So it's quite common to have more genomes than patients from that kind of sampling.
Yeah.
Okay, so the next question is from Anne. She says, thanks for the great overview. What do you think are the biggest hurdles so far for more global molecular surveillance? And also, what made the UK so effective with their approach?
I think probably governments and politics got in the way of a lot of this. Some governments don't want to share data because they fear to be punished. You can actually see, unfortunately, South Africa were effectively punished for their great surveillance work and for making their data available for both the Beta variant and for Omicron. So that is a big hurdle, and it's quite difficult. We are involved in a lot of different international sequencing projects, and that is a constant stumbling block, and it can take months to set up sequencing projects with other countries. And it's usually the paperwork aspect. And even then, governments often want to know first if there's anything, potentially bad in there, and they can either suppress the data or, massage it or something like that. So that is the biggest hurdle, and I don't think it's one we're going to solve necessarily. But what made the UK so effective was just we're told, go ahead and just do it without interference. And we just went ahead and did it. And, Nick was in those early stages, so I'm sure he can give more information on that.
Well, we definitely took the view that the data should flow, and as quickly as possible. And I think, as you say, it almost doesn't get vetted or controlled. It goes straight up into the public databases, and we also have a broad network of UK academics and public health scientists looking at that data in parallel. So I think that's another key point. It's not just the provisioning of the data, it's the ability for lots of people to also analyze it, and I think that's a sometimes overlooked point. So it's there for the international community, but it's also there for a lot of different scientists to dig into, and that's generated a lot of different types of studies and a lot of different types of insights. Okay, so quickly, we have 3 questions from Lucas, 3 related questions. So when investigating an outbreak, do you sequence all the collected samples, or do you do some kind of surveillance sampling? Is there a limit of the number of mutations between the sequences to classify them as related? And I guess the follow-on question from that is, would it be acceptable to investigate an outbreak with partial sequencing? So, for example, I guess Sanger sequencing of a single amplicon.
Okay, so the first one, do we sequence everything? Or in an outbreak, we would try and sequence everything if the outbreak isn't too big. But if it is a very big outbreak, say in a closed environment, you expect everything to be the same. So in some cases, we did, put a practical limit just on capacity. But that's just down to you just have to look at what you can do at the time. And say in the case of a factory where you have hundreds of positives, we just took a random sample. I think it was whatever would fit on an nanopore run, and then they're all the same. But it's in the same kind of vein, when we did every single case in an outbreak in a different factory, we were able to tell that there's multiple introductions because we'd sequenced everyone. And we also found that there was one random other lineage that popped in from the community. So, you get different levels of information depending on how much you sequence. But generally, for infection control, they probably just want a few samples just to confirm it's all the same. Is there a limit on mutations classified as an outbreak? Well, SARS-CoV-2, it mutates very slowly, generally. And so often, if it is an outbreak, they'll all be the same, or they might have one SNP. And anything more than that, it's probably not very closely related, unless it's a single event that has caused multiple mutations. And it'll vary depending on the other information you have. You can't just rely on genomes alone. You have to add in all the other epidemiological information to actually give you insights. And also then you have to look at the community as well. If something is spreading massively in the community, it's not an outbreak, you're just seeing the community. Okay, and then lastly, Sanger-based sequencing. If you've no other options, sure, it'll be okay. Some information is better than nothing, but it is not really used in the UK for surveillance. It's all, either Illumina or Nanopore. I know some countries early on did do a lot of Sanger sequencing, and they then didn't build up their infrastructure and have really regretted it. Other countries take the approach where they'll do Sanger sequencing just to get quick results while the actual proper sequencing on Nanopore is going on. So it really depends on what's available. But I would say, it's quite cheap to do, say, high throughput Illumina sequencing. It costs, it's probably down to about £5 if you do huge batches. And Nanopore is quite cheap as well, and quick, and you don't have the infrastructure costs as well. So Sanger sequencing probably isn't that cheaper than you think, but if it's the only machine you have, well, then use what you've got. Okay, so I think we'll call it a day there.
Thank you very much, Andrew. That was excellent, and thank you also for the excellent question.